Ahmed. I'm a graduate student in Dr. Ben Langmead's lab, and I'll be presenting work today from Johns Hopkins, University of Florida, and Dalhousie University. And as mentioned, the title of my talk is Pangenomic Matching Statistics for Targeted Nanopore Sequencing. So nanopore sequencing is a long-read technology that works essentially by passing a strand of DNA through a pore, and by monitoring the change in current through that pore, you can sort of decode that sequence into a sequence of bases that pass through. And one of the exciting aspects of this technology is it allows users to perform targeted sequencing entirely in software, so avoiding experimental protocols, which could be sort of time-consuming and difficult at times. So essentially how it works is that as you're monitoring the signal, if it looks like a sequence of bases you're not interested in it, you can just reverse it out of the pore and eject it. On the flip side, if it looks like a sequence of bases that you're interested in, so based off of your study, if it looks like a sequence of interest, you can just let that read essentially keep sequencing and you get the full sequence. So that's essentially how it works. Now, one thing to keep in mind about nanopore sequencers is that you just don't have one pore sequencing at one time. You have numerous pores. So for example, one of the smaller sequencers that nanopore have called a MinION has 512 pores sequencing at the same time. So you have a lot of simultaneous streams of data. So if you want to accomplish this with software and practice, the software needs to be fast enough to keep up with the incoming signal, essentially to kind of make sure you're doing it on time and able to keep up. So there have been methods in the past that have accomplished this, namely Uncalled and Refish have been able to perform this in practice. And there are actually additional tools as well, such as SquiggleNet, which we'll hear more about later today. So I just wanted to highlight one result to kind of show you guys what these methods allow you to accomplish. So in the Uncalled paper, they kind of showed they had an experiment where they wanted to sequence a mock community. So the mock community consisted of yeast as well as 7 microbial species. So the goal was to target the yeast and eject all the microbial reads. So if I show you a figure here, it's just really basically we want to kind of look at these 2 bars. And we can see that Uncalled essentially allows you to have higher coverages of your target sequence, which was yeast, and a higher % yield. So overall, this is kind of the goal of these types of technologies, to give you higher coverages for your sequences of interest, which will help in downstream applications. And I wanted to highlight this paper because it kind of points to the motivation for our work. So in the paper for Uncalled, they mentioned that performance degrades as references become large and more repetitive. So it kind of pushes the need for faster methods to classify against large repetitive databases. So you may ask, why is this important? Why can't we just use kind of smaller files? So I'll kind of give 2 reasons, and then we'll kind of talk about them later on in the results. So the first one is that it's going to help you to improve your targeting accuracy. So we'll see that in the results later on. And the second reason is that it allows you to target genomes that aren't necessarily explicitly in your reference. And again, we'll see that kind of an example of that in the results. So now kind of doing a method overview. So our method is called SPUMONI, which stands for Streaming Pseudo MONI. So MONI is actually a tool that's published in RECOMB this year, and Max Rossi will be giving a talk on that next Tuesday. So I encourage you guys to check that out. So what SPUMONI focuses on is making rapid targeting decisions based on an input database. So essentially making a binary classification for each read, whether it appears to be matching to the database. And the key intuition that it uses to make these decisions is using a read's matching statistics or pseudo matching lengths, which we'll talk about on the next couple of slides. So using those with respect to a reference, and then see if it, usually tell you if there's a good approximate match to the reference. And then what allows SPUMONI to be able to handle these large repetitive collections is the use of the R index. So the R index is a data structure that has an O of R space complexity, where R is the number of runs in the Burroughs-Wheeler transform of the text. And the key idea for large repetitive references is that typically R grows sublinearly with respect to the reference. So that's kind of where all the efficient indexing comes from. And then I just wanted to highlight kind of 2 key areas of SPUMONI that extends upon MONI. So the first one is that SPUMONI adds a null index as well as a testing framework to be able to distinguish what's a significant match to the reference. And it also replaces MONI's 2-pass matching statistic algorithm with a faster streaming algorithm. And it calculates a new quantity, which we're calling pseudo matching lengths. So I just wanted to highlight some of the strengths of the R index to kind of make sure we kind of understand what it's doing for us. So what is the R index first off? So it's kind of the combination first of Mackinnon and Navarro's O of R rank data structure over the BWT. So this is a compressed data structure And Gaggi et al.'s OVAR suffix array sampling for the, so these two ideas kind of combine to make the R index. So this combination allows you to do efficient queries to count the number of occurrences of a pattern in a text, as well as locate those occurrences. And I kind of just wanted to highlight here one of the results from the MONIT et al. Work. So it's showing you here the peak index memory as we index larger and larger collections of human assemblies. And you can think of this peak index memory as being similar to the size of the index. So you can see here this sublinear aspect. So this sublinear aspect is sort of a direct experimental result, just showing you how R is sublinear as the sequence gets bigger. So that's kind of what explains this sublinear aspect of the curve here. So now I want to talk a little about matching statistics. So matching statistics for a pattern with respect to a text is an array of length m, where m is the length of the pattern. And then each position in the array corresponds to the longest prefix of the pattern starting at position I that occurs exactly in the text. So a less formal way to think about it is that they're half mems, or maximal exact matches. Because essentially what we're interested in is starting at a certain position in a pattern and asking how far can we extend our exact match in one direction that still occurs in the pattern. So here's an example to kind of wrap our heads around this quantity. So for position 0, the matching statistic is 3, because you start at position 0, you go to the right, you see that TAC occurs in the text exactly, but TACG does not. So that's why this is a 3. The next one, we start at position 1, we keep going to the right, we see that ACGG occurs in the text, but not ACGGT. So then let's kind of fill in the next couple ones here. And we can see here that this one is a 2, so TA occurs exactly in the text, and the last one's 1. So this is kind of just a basic overview of matching statistics. So now that we understand matching statistics, I wanted to kind of show you the basic idea for MONIT's algorithm and then compare that with SPUMONI to kind of understand where we're coming from with this new quantity. So MONIT uses a 2-pass algorithm to calculate matching statistics. And in the query loop, there are sort of 2 cases that each base in the read can fall into. The first case is that the current pattern, the character in the pattern equals the current character in the BWT. So if that's the case, that means that we can extend the current match we have by 1. The second case falls into 2 subcases. So when the characters are not equal to each other, we can either still extend it, but we just have to sort of jump to another row in the Burroughs-Wheeler matrix, or we can't extend it, but we can possibly retain some of the match. So let me kind of show you what these cases mean in terms of the matching statistics. So we're going to start at the end of the read. So we're in case 1, so the black box here. So we add 1. We're in case 1 again. We add 1 again. Case 1. Then we hit case 2A. So this means we can still extend by 1. So that's why this becomes a 4. Case 1. And then we hit a case 2B. So this is a case where we can't extend their current match, but we can possibly retain some of it. So that's why this value kind of dropped down to some value less than or equal to 5. And I'll kind of zoom through a little faster through the rest of these. So we had case 2A, case 1 again, case 2A, and case 2B. So essentially what I'm showing you here is how each of these cases affects the matching statistics. So now let's talk about SPUMONI. So SPUMONI is an online algorithm. So it's a 1-pass algorithm, and it calculates a new quantity, which we are calling pseudo matching lengths. So the first case is the same as MONI's case, but the second case is where they differ. So if the 2 characters, the current character in the pattern does not equal the current character in the BWT, then we just set the length to 0. So essentially SPUMONI is not trying to distinguish between these 2 cases, and we're just going to say we're going to set it to 0. So let's look at the same read and see how that affects kind of the pseudo matching lengths. So we start out with case 1. So we add 1, case 1 again, we add 1, case 1 again, and then we hit a case 2. So that's we set it back to 0. Now case 1, and then when we case 2, we hit 0, 0. Case 1, we add 1, and in case 2, we go back down to 0. So now that we have the pseudo matching lengths and the matching statistic for both of these reads, the same read, we can kind of compare them visually, and we can see that one of the effects that pseudo matching lengths has is that overall it lowers kind of all these values, but it also has this effect where it kind of squashes some of these peaks. So you can see how it kind of squashed this peak a little more than this one. So this is a simplification because we are not trying to distinguish it. So this simplification allows us to throw out some parts of the index of MONI, which allows us to use less memory and to be faster. And the question you might have is, how do these two quantities, how do they help us when it comes to classification? Is one better than the other? So I'll leave that as an open question now, and we'll actually be able to answer it when we go to the results. So here's sort of a high-level view of what SPUMONI does. So we're starting out with a stream of base calls, kind of like a nanopore sequencer. So you can imagine each pore is kind of giving us a read. And what we do is we generate the matching statistics or pseudo matching links against a positive and a null database. So the positive database is essentially your sequences of interest. So whatever you're interested in. And the null database is just a reverse of that sequence. So we generate with respect to both of these databases, we're going to get two distributions. And then we compare them using kind of a simple statistical test, the Kolmogorov-Smirnov test, to kind of compare them and see if they're significantly different. If they're different, that means that the read is matching to some sequence in the positive database. If they're not, then it's not really matching. So that's how we make our sort of ejection decisions. So now kind of transitioning into some of the results. So the first question we kind of had is, how does sequencing error affect SPUMONI's classification? So with long-read technologies, you're typically dealing with higher error rates. So it's kind of a natural question to ask. So we use kind of the mock community example, where we built an index for the microbial species in mock community, which includes E. coli. And then we looked at the matching statistics for E. coli reads as we changed the read accuracy. So kind of going from the low end to kind of the high end. And if you look at the blue distributions, so these are the distributions of matching statistics from the positive database, you can see that they're starting to flatten out and kind of getting pulled to the right as you kind of get higher and higher accuracy. So this makes sense because you have less sequencing error. So you have less sequencing error that could be affecting the half mems. So that's why it's getting pulled farther and farther to the right. And overall, these two distributions are getting much different from each other. So you're going to assume that the classification is going to improve as accuracy improves, which is what we see with the actual numbers. So when we look at some of the actual classification accuracies, we can see that SPUMONI's accuracy is quite high, 99.5% or greater for reads that have a 99% accuracy or higher. So that's really good. Even at 85% accuracy, SPUMONI's accuracy is still relatively high. It is behind Minimap2's, but overall for reads that have a typical nanopore error rate, the kind of the normal range, SPUMONI does quite well. So now kind of transitioning into another experiment. So the question we had with this one is, can using a pan-genome reference allow us to target a strain that is not in the reference? And how does it compare on time and memory? So here, kind of the gold standard we're comparing with is Minimap2. And then for this experiment, we use real mock community reads from the uncalled paper. So we kind of had the same goal. We wanted to target the yeast reads and eject all the microbial reads. So first, I kind of just want to highlight the two different types of databases that we used. So we use a one genome reference. So this means that we're taking one random strain from each of the mock community species, the microbial mock community species, and it doesn't include the exact strain. And the pan-genome reference is when we include one reference or all the strains for all of the microbial species, but not including the exact one. And then the three approaches we benchmarked were SPUMONI MS. So this is using matching statistics. SPUMONI is using pseudo matching links. And then Minimap2 is sort of the gold standard here, alignment approach, similar to what Reedfish uses. So when we look at the accuracy when using the one genome reference, we can see that SPUMONI is slightly behind Minimap2, but it is ahead of SPUMONI MS. So this is kind of the trend we saw with all of our experiments, is that using pseudo matching links has a better effect, or gives you more power for when it comes to the binary classification. But overall, when you look at these numbers, they're a little lower than what you would hope for when it comes to kind of a simple binary classification task. But when you look at the pan-genome reference, you can see that overall, the numbers do quite well. So it kind of shows you the hit you take when you choose a random strain to do your targeting sequencing with. But again, when you look at the pan-genome reference, you can see that SPUMONI again is slightly behind Minimap2, but the numbers are kind of comparable. But the big difference comes when you look at the time and memory. So you can see that SPUMONI uses 4 times less memory, an index that's 16 times smaller, and 12 times faster. So this is kind of highlighting the strength of SPUMONI when it's indexing large repetitive collections. So to kind of answer the questions above, yes, using a pan-genome reference allowed us to target the Zymo mock community strains, even though they weren't in the pan-genome reference. And we're able to do it using less memory and faster than Minimap2 with similar classification metrics. Okay, so now transitioning to another result. So here we have the question, can SPUMONI extend to other scenarios, in particular when we're using human genomes? So this is sort of modeling a human microbiome experiment where you might want to study the microbiome reads and you want to eject the human reads. So we built an index over 3 human genomes. And then we looked at the matching statistics for human and bacteria. So for human, you can see that the positive distribution, it's stretching to the right because it's matching with the human index, but the bacteria reads aren't really matching, so the distributions aren't really diverging from each other. So if we look at some of the classification metrics, we can see that both SPUMONI and MiniMap2 are quite high and close to each other. But again, the big difference comes when you look at SPUMONI speed, and it's 2 times faster than MiniMap2. Again, kind of showing the strength of the R index for large repetitive collections. So now kind of in summary, as well as adding in some more points. So we discussed SPUMONI, which is a streaming algorithm for targeted nanopore sequencing. It uses matching statistics or pseudo matching links to classify. And the real strength of it comes from the sublinear aspect of R. As you add in more repetitive sequence, you're able to get this sublinear aspect, which helps the R index to be efficient for large repetitive collections. We also talked about SPUMONI's computational efficiency when it comes to pangenomic references in particular. And we started the talk by talking about the numerous channels, so you have to be computationally efficient. And to add on to that, nanopore has a lot of portable sequencers like the MinION and Fongle, which can be connected to things like laptops. So it's good to be as efficient as possible when you're using that type of hardware. We also talked about SPUMONI, how it opens the door to doing targeted sequencing for strains that are not in the reference, either because they haven't been assembled or they can't be cultured. So this potentially opens the door to doing assembling a lot of strains that couldn't have been assembled before. And also an important point I wanted to highlight is that SPUMONI is not limited by the use of a K-mer length setting or sort of a simple threshold over, for example, over the length of a mem. And the reason for that is because using the null distribution allows our notion of significance to be a function of the database size and the database sequences, as well as the error rate. Because you can imagine with a large database, the length of the random match is going to be higher than with a smaller database. So it's important to have a function of significance that can adapt. And future work will consist of implementing the needed software for SPUMONI to actually interact with the read until API directly to actually perform the targeted sequencing. So thank you for listening. Thank you to all our collaborators, and thank you to all the grad students and postdocs in the associated lab.
Thank you, Omar. We have 2 questions from the audience. The first question is from Camille. The question is, great talk, Omar, thank you. What are the bottlenecks of SPUMONI, and/or where do you see future developments occurring?
Yeah, that's a good question. So we spent a lot of time recently kind of benchmarking SPUMONI. So, one bottleneck could be with the data structure, but I think we've really optimized that. And there's actually future work done by some other students that actually will help to optimize the R index to make it faster. But also we've been looking in future development of trying to just see if we can index smaller sequences using kind of sketching approaches to help us speed up the actual query process. So that's kind of one potential for future development to help speed up the actual underlying query.
That sounds good. Another question from Bara Orabi. Great talk. How does the tool speed performs for the median read? Is it fast enough to reject the read in real time?
Yeah, so I didn't really get a chance to mention this during the talk, but so what we did for the experiments is that we just focused on the first 720 bases of the read, sort of 1.6 seconds, because that was sort of a time window that ReadFish sort of mentioned in their paper that they could eject most of the reads by. So we were essentially faster than MiniMap2 and sort of making those classifications. So our hypothesis is that since we're faster than MiniMap2, we should be able to, when we transition into a real setting, be able to eject in real time since the reads are long. And if we can make decisions within the first 720 bases, it makes us confident that we can actually eject the read in real time.
Okay, that sounds good. There's another question, but for the interest of time, I would suggest that you type it offline.
Okay.
And that we move on to the next talk. So thanks again.